So, hi everyone, welcome to episode 2 of Poly Demo Days. My name is Maya Stedman, and I'm a bioinformatics scientist here at Elucidata.
Hi everyone, I'm Kritika Gaukar. I'm a senior solutions architect at Elucidata, and happy to see everyone here today.
Yeah, for those of you who aren't familiar or haven't been to a demo day before, Poly Demo Days are episodes where we'll feature demos and technical deep dives into biomedical data integration and analysis. You can expect to learn about new features and capabilities, as well as what we over here at Elucidata are really excited about in the world of data science. Today's session, we're going to demonstrate what ML-Ready Flow Cytometry Data looks like on Poly and how it can be used on our platform. And for this conversation, we've even have a special guest for this episode, our resident flow expert, Marianne Santaguida. Marianne is a trained biologist. She trained at McGill and then later at UCSF. Soon after, she was at STEM-Centrics and was leading their platform to identify and validate drug targets on oncology. While there, she leveraged different forms of biomedical data, including flow, microarray, omics data, and used all of those different data types to drive their platform forward. A few jobs later, she became a VP partner at Notable Labs, which is a clinical stage precision medicine company, and they used highly automated flow cytometry platforms to discover insights on AML patients. We're really glad to have her today. She currently is advising startups like Elucidata on how to use their data, especially flow data, and how to handle, work with AML data and help any company that's looking for cancer insights. And today she has graciously offered to guide us through some of the best practices on using flow cytometry data and some of the challenges that we face making flow data usable for analysis. So that was a long intro. Welcome, Marianne. We're really excited to have you here.
Thank you, Maya. It's a pleasure to be here.
Okay, so let's jump right to it. In our last episode, we can go to the next slide here, Shruti, thanks. In our last episode, we showed how to explore Polyomics Atlas, either through which Polyomics Atlas is the term we use for our, it's what houses our curated data on our platform. It's our platform. So if you're not familiar with the terminology, when I say Polyomics Atlas, that's what we're referring to. We showed you how to navigate it either on our UI or through a programmatic interface. And we showed you how to search for and find relevant data sets on Poly. It's super easy because we've already reconciled the differences in formats and ontologies and filled in the gaps that are there in metadata by applying our two-step curation process. So the first step is that we convert all of our standardized data into a tabular schema, and then we harmonize the metadata. So if you're not familiar with that term, we'll talk about it a lot. The metadata is what describes the data set or the sample. And we've taken that metadata and we've actually linked it to the physical data matrix. So they're always together. And so now that all the data on Poly has been curated, it's ready for machine learning and bioinformatics applications. So that's our recap of our first episode. And if we go to the next slide, we're going to jump into this episode. And what we want to really emphasize before we get started is the wide variety of data types that are hosted on Poly. There's more than 20 different data types on our platform. It includes omics data like transcriptomics and metabolomics, as well as non-omics data, such as lab measurements, which could be like blood glucose levels or oxygen consumption rates that were measured, as well as plenty of flow data, as we'll get into a little bit later. And so it's easy to see how we could take omics data sets, which are already in a structured or a matrix format, and harmonize those. But we've also applied this same level of curation and that same curation process to unstructured and semi-structured data sets as well, making data that was at one point only usable by human experts ready for analysis in a bioinformatics pipeline. And that's what we're going to highlight today. So we want to take a deep dive into flow cytometry specifically and discuss some of the best practices for both generating flow data, but then also using it in your research and some of the challenges that we face making flow data fair. At this point, I do have to admit that when we first started working on this episode, I knew nothing about flow cytometry. And yeah,
I
so
like,
this has been, go ahead, Kritika.
I just known that it is something that you would use to characterize cells, but didn't know anything more than that, like what the data is, what do we study from it. So it was
Yeah.
a great learning experience talking to Marianne before.
Yeah, absolutely. So after we've been chatting with her, now I'm a full believer in the importance of flow data for biomedical research. And I'm really excited to see how these data are going to be used for the benefit of patients in the future. So on that note, let's start at the beginning with a little bit of Flow 101. And we're trying to get at what exactly is flow cytometry. So what are we actually measuring when we say that we're going to do a flow analysis?
So flow cytometry is a type of proteomics. We're typically taking single cells and staining them with antibodies that detect proteins on the surface of these cells. And these antibodies are linked to a fluorescent tag that allows us to detect it. So we take the single cells, we march them down a single file using hydrodynamic focusing, and then we use lasers to excite those tags, detect the different proteins on the surface. And then there's a bunch of very complex electronics that detect those signals and computate that into something that we can visualize through the software provided with the machine. So typically we're using this technique to look at different protein populations, the overlap of these surface markers, and how they compare to other single cells in the sample. So you could look at CD4 counting, which is a marker on the surface of an immune cell called the T cell, HLA typing, which would be important in organ donation, or immunophenotyping, which could be something that we could be doing in cell therapies.
Nice. And so when talking about if someone wanted to bring flow cytometry into their lab, what sort of, is this a really difficult procedure to perform? What kind of skill and training is necessary to run and analyze flow data?
So typically, flow, the complexity of flow is dependent on the complexity of the number of antigens that you're looking for. Most analysts go through training, depending if they're doing it from a research setting or from a clinical setting. They'll either, on the clinical side, they need to be certified. And the complexity is not only in staining the cells and running them through a machine, usually with the help of an operator, but in the analysis on the back end. So as you as you increase the number of antigens that you're detecting, the fluorescence, the signal of those antigens detected, and the overlap of that detection can add complexity. And deconvoluting that is called compensation, and does also require skill.
Yeah. I have, as we've been talking about this, I've learned so much about what goes into it, and all of the different controls that you need to have in place to make sure that the data is usable, as well as the effort that's put into the data quality itself, right? And thinking about who is generating this data. Before we move on to the next slide, just sort of one more background piece of information. Let's talk a little bit about the data quality associated with flow. So I know it comes from a lot of different sources, but how much of a role does the skill of the analyst and sort of the who is generating the data play into what you're looking at when you're analyzing the data?
Well, it can be very important, not only in the type of controls that you're using that allow for the analysis and the deconvolution of the compensation, but on the clinical side, The analyst is a certified flow operator in order to run that sort of diagnosis for patients. The actual flow cytometer is approved for clinical analysis. And then the actual analysis and the interpretation is done by a pathologist that would report that information back to either the clinician or an oncologist who is the doctor requesting the information.
That's great. So it kind of leads us right into our next topic for background information. And if we could just go to the next slide here, Shruti. Awesome. Is I wanted to talk a little bit more about the different applications. So we've talked about what it is, and now let's talk a little bit about where it's coming from. So who is generating it and why are we generating it? What are some of the applications for flow cytometry data?
Well, there are both sides on the research and development side, and then on the clinical side, there's a lot of applications to flow cytometry. On the research and development side, we often use flow cytometry to look for antigens that could be the targets of new therapies. It could be for the identification of biomarkers so that we determine a population of patients that either respond to a therapy or that require different treatment. Immune monitoring and vaccine development. This is really critical in understanding before vaccines go into patients and in clinical trials. Are patients responding to the vaccine? Are they eliciting an immune response that would allow patients in the future to be protected from the vaccine? And then cell function and monitoring. We use this in a lot of different applications, both on the research side and on the clinical side, to look at either the response to cell therapies or the development of different drugs in different animal patients or animal studies.
And for response to immune therapy or like vaccines, what are we like trying to interpret from flow cytometry?
So typically in vaccine development, initially we're looking to see over time if the vaccine generates a B cell response. This is the cell in the immune system that would be responsible for the protecting initially the animal model and then the patient from the vaccine. So to see if there's a response, and then to see if they're with the virus present, if there is a response to that vaccine.
That's great. And then, sorry, Kritika, I didn't mean to cut you off.
Oh, I was just going to say that's quite interesting, just because we've been like hearing so much about vaccines and immune responses. That's useful information.
Yeah, I wanted to kind of hone back in on kind of the difference between the clinical and the research applications for flow cytometry, right? So we can see that there would be pluses and minuses to ICE, I think, or to either application. I think you had mentioned previously that there's actually a set of guidelines that the clinical or people who use flow in a clinical setting have to follow. Could you talk a little bit more about sort of the pluses and minuses about using flow data and generating it in the research applications versus a clinical application?
So on the clinical side, the analysis is much more rigid, right? And in order to provide diagnosis for patients, there are specific guidelines that are listed not only in protocols and being approved. Usually you have is what's called a CLIA lab. So the laboratory itself has a specific designation, the equipment and the operator, but those guidelines are very rigid. Even the antibodies that are used for those detection. On the research side, there aren't really that many rules. We have a lot more flexibility, which allows for a lot more complexity. And this can be very interesting because we can do a deeper dive into what's going on, use a lot more surface markers, and really characterize things from a different level. I think both sides are really important to how we look at biomedical data and how we do research for therapies, and they have their strong points and their disadvantages as well.
Yeah, I could definitely see that, where like the flexibility on the research side, you know, coming from the startup and biotech realm, like that's where I'm used to seeing flow data. And I was so surprised to see how much structure was involved in collecting flow cytometry data for the clinical setting. Surprised and relieved, I guess I should say. So it's great to see it. And then so I wanted to again just drill down one more layer deep, and I was hoping that you would actually walk us through some flow plots. So these are, if I jump to the next slide here. Here we go. So yeah, if you could just walk us through here, what we're looking at is, I believe, these are patients and control samples and this is their flow scatter plots. And if you could, you know, with all your experience, sort of walk us through what we're looking at and how you can use this information on these plots to determine whether or not a patient would be diagnosed with AML.
So in this case, we're looking at one patient that has normal bone marrow and 3 patients that have been diagnosed with acute myeloid leukemia, or AML. And we're looking at light, how the cells, their complexity, which is called side scatter. And on the x-axis, we're looking at CD45, which is a marker of hematopoietic cells. So normal patients have a very typical profile of their bone marrow, and it's fairly consistent from patient to patient. And we see the little blue circle is got a few dots in there. Each dot represents one cell, and we don't see too many. In the 3 patients with acute myeloid leukemia, we see that that blue population is now being pointed to with an arrow is much larger, and this represents the cancer cell, which in acute myeloid leukemia is called a blast cell, a population of cells that shouldn't be present in the bone marrow. We also see that the normal populations, the other populations that should be present, really look different. And so there's a skewing of the cells to generate this blast population. Typically, in clinical flow, we would go on to characterize that blast cell with additional markers to really understand what's on the surface of that cell. And it would also be combined with some omics information, usually genomics, to look for mutation analysis. And it's the combination of that information that would be used for the treatment of the patient.
So this is just the beginning. These first diagnostic plots are really just the start of the data collection for these patients.
Right, yeah. But probably the most important place to start is identifying a cancer population that isn't normal.
Yeah. And then just to get a little bit clearer, so are these, I believe these are bone marrow samples, but you see a lot of flow cytometry also done on blood as well in patients. So you could expect to see many sample types from the same patient.
That's right. So typically you would use the sample where you expect the disease. So acute myeloid leukemia is typically a bone marrow disease. And so bone marrow is used as the diagnostic sample. But in acute myeloid leukemia, it does spill over into the peripheral blood, not always, but typically. And often they'll use peripheral blood to monitor patients over time because bone marrow samples can be pretty invasive.
Yeah, I've heard that the procedure is very unpleasant and can be quite taxing for the person who undergoes it. One other thing that I kind of noticed while looking at these plots, and Kritika, if you have any other questions, don't let me take over from you here. But one of the things that really struck me when we were talking about these is how different each one of the AML patients' flow plots look, right? This isn't something that I could look at and know without being told that, excuse me, just because I'm completely untrained, really that these were all the same population, or the same, not population, but the same diagnosis, right? And it kind of struck me because this is something that a human expert has to look at these plots. This isn't like, oh, you got a checkbox in this quadrant and therefore, you could easily convert this image into something that can be machine readable, right? You need that human intervention to gather the information and to understand what we're looking at here.
So traditionally, flow cytometry, you're right, there is training and an expertise that goes into the analysis. And I think over time, that will move to a more machine learning and automated analysis because of the quantity of samples that are available to allow for that.
And I was just going to follow up with, once you have the diagnosis, would we also use flow cytometry to track the patient if we're given them, some immune therapy to see the response?
Absolutely. So they do use a clinical flow cytometry to determine the response of the patient. So this population with the arrow, when patients are receiving therapy, should decrease over time. Typically, patients receive multiple cycles, and in between cycles, they will do an analysis. They also use this sort of clinical flow cytometry and immune monitoring to look at the residual disease in the patient and to make sure that the patient doesn't relapse, and to track their relapse over time, when to start treatment again or to try to start a different treatment.
And would we also document, like percentage or frequencies of the cells that shouldn't be, so dense in AML, so that we can record, the profile of a patient just to get numbers,
Absolutely. We would record two things. The overlap of the markers used in the analysis to look at populations, and then also to look at the level of those individual markers on the surface of every cell.
So I have sorry, one more question just to clarify this, because this is, I think, a really interesting point. Typically, when we're having when we report this, is the doctor's not seeing the flow plot, or the clinician isn't seeing the actual plot, they're seeing a report that has values on it?
They for clinical flow analysis, they'll receive both. They'll receive
both.
the
Okay.
number value, they'll receive a summary, and they'll also receive the flow plots, pictures.
Yeah, so it's a lot of information for every patient. But I think, Kritika, we wanted to move on and talk a little bit about the underlying data. So when we talk about those values that are being reported, the information that we're actually, I think you had mentioned there's two things, the fluorescence and the population that are being reported. So let's talk a little bit about the move away from the images and talk a bit about those values themselves and how we can, what we can do with them.
Yeah, so we talked a lot about, the variety of applications, and we also heard Marianne talk about, there'll be multiple rounds of flow cytometry data per patient. So a lot of data is being generated, that is flow cytometry. And a lot of it is available in public repositories. One of them is import. And I just wanted to emphasize that our infrastructure supports multiple types of omics data. For example, transcriptomics from Geo, and then we have gene dependency from DepMap, and flow cytometry data from import. And all of that resides in a central repository, which we are calling Omics Atlas. And through that, you would be able to access the data. If you're a biologist, you would look at, the UI, the web apps that we have. And if you're a bioinformatician or ML researcher, you'll be able to access the data programmatically on or outside Poly. And similarly, if you are generating a lot of flow cytometry data, and you're also profiling the patient's gene expression or mutation data, all of that can be curated and be accessible, similar to what we've done to public data. And internally in your company, you would be able to have a lot of people reuse the data and just make the data accessible and findable throughout your organization. And a lot of us kind of know or have read literature where we've used omics data to derive new insights or generate some hypotheses. But I wonder if there are challenges in reusing flow cytometry data.
Yeah, I think you had mentioned, that the clinician or anything, there's 3 actual files that get sent along with any piece of flow cytometry data. So, I mean, just thinking about keeping track of all of those files would be it would be a real challenge. Would you say that it's limiting in our ability to reuse flow data for other applications?
Absolutely. The flow data is typically stored as images such as JPEGs in Excel spreadsheets, and individual patients are stored separately. And so this information can be difficult to incorporate, to go back to and use for future analysis, but probably more importantly, to incorporate with other omics information, such as genomics or transcriptional information.
Yeah, so I think that's a great segue to talk about the data schema that we have on Poly. So like Marian said, the data can exist in so many different ways. We've seen that in public repositories as well. Some data sets are TXT files, some are FASTQ files, some where the metadata is in the supplementary. So data can be, difficult. But what we are trying to do in Poly is to standardize the data so that it's accessible in the fastest way possible. And you will be able to query it because we have some ontologies that we use to annotate our data sets. For example, if you're looking for AML, all of our data sets that are AML related would be annotated with the exact term that mesh ontology uses to describe AML. And if you're trying to access, just human data across multiple repositories, you'll be able to search for those data sets by just using, organism equal to Homo sapiens, because we have a curation process to annotate the organisms across all the repositories and all the data types, just making it more usable, and standardized. And the data itself, which is what we call the data matrix, usually omics data is in some matrix form. That is saved in GCD files, and we'll talk briefly about the actual format. But the GCD file allows you to store the data matrix along with the associated metadata in one file. So you don't have to look for, the data and the metadata separately and then merge, the associated data to do some quick visualization. The whole data package has metadata and the data matrix, so you can do pretty quick visualizations or interpretation of the data. And for some data sets that are more complex or, large, like single cell, we store them in H5AD because it's a popularly used format for single cell, and a lot of tools are able to use H5AD. Again, the data and the metadata are all saved in one file, easy and accessible to use. Let's look at
add on. Sorry, go ahead, Kritika. I didn't mean to cut you off.
No, you should go ahead.
Okay, I was just going to add, just re-emphasize that this level of curation, you mentioned a lot, like the data set and the sample level, but we also apply it to the feature level. So the actual row level information is also harmonized. So you're not limited by searching by disease or by some other phenotypic description of your sample or your data set. You can actually look for your genes of interest, your targets of interest, at the feature level, which I think is pretty unique and really useful.
That's a great point. Yeah. Yeah, so let's look at the GCD format a little bit in the next slide. So Maya was talking about how we've also curated the feature level, which is the gene level information for transcriptomics data. And for example, if you would want to search for, the gene expression for this FOXR1 gene across multiple transcriptomics data sets, you'd be able to query specifically for expression data for that gene, because we have curated the row metadata, which is here, and you'll be easily able to query this gene across multiple repositories and multiple diseases and organisms to, derive insights from it. And we also have the sample metadata, which is usually the columns in the data matrix. And this data has, some sample specific information like the gender, disease, tissues, etc. And we've done this for transcriptomics data, and just wanted to reiterate that because it's standardized, we could query across multiple repositories like Geo, cBioportal, TCGA. But we've also done that for analyzed FCS files. In the next slide, we'll see how the FCS files are stored. And again, you see the three components: the sample level metadata with the sample related metadata, the row metadata, which has the actual population name reported, and also something specific about it, like what are the units used, because there can be multiple units that you can report from flow cytometry data. And the matrix itself is reporting the frequency of each gene population, of each cell population. And since it's formatted in a similar way across all the patients in the study, you would be able to merge those files together as well, which is what we are going to see in our notebook demo. So I can just start sharing my screen.
I just think that it was really great to see how we're taking these data files, these unstructured data files, and by putting them in the structured format, it's important to keep that in mind when we're looking at Kritika's notebook, when the experiment that she's going to walk us through, how empowering that structure is in applying it to an analysis.
Yeah, so this is our notebook. And in our last episode, we did cover some parts of it. So I'll just skim through it, but you can reach out to us or go through our YouTube video from episode 1 to know more about those capabilities. So just to start out, PolyPython is our library that provides convenient access to our curated data. And it's very easy to install because it's just a Python package. You would do pip3 install PolyPython, and it'll be ready for you to use either on Poly or in your local environment or wherever you do your data analysis. So I'm just going to run this cell to import all the libraries that are required for my analysis. And I will add my authentication. Like each user has to get their authentication from our Poly platform. And I've already done that. So now I am able to access all the data repositories that we have on Poly. And this is just a brief look at the metadata field that we talked about previously. So you can see a lot of metadata is curated and follows specific ontologies at dataset level and at sample level, which allows us to search for these data sets conveniently using SQL. And I'm just going to run that. And it will go through geo and import, mainly because I'm trying to identify data sets related to influenza, where the KW data type is transcriptomics, which is what we'll have from geo, or it's cytometry, which we will find in our import repository. And it's done a quick search, and we have 374 data sets from geo that are related to influenza, and 126 data sets from import related to cytometry, which is great. That's a lot of data. But for today's demo, I identified a study that I felt was really interesting. It's a longitudinal study for a vaccine administered on healthy children. Actually, 2 vaccines administered in healthy children. And they had flow cytometry data to characterize the B cell population, like Marian talked about how that's useful in vaccine development and identifying the response to vaccine. And we also have the transcriptional data in geo, which helps identify the changes in expression in different time points in immune-related genes in patients using one vaccine over the other. So I feel like it's a great study to get an overview of the patients or like the children who have the vaccine. So let's explore some of the data quickly. So here is the GSE ID for my geo data set. And I am just going to quickly download that file by just running download data and giving the repository name here. And it's already downloaded. It was quite fast. And you are able to parse the GCT file using the parse function from CMapPy, which is a publicly available Python module, which allows us to parse our GCT file in the 3 components that we've shown before in our slides. So the count matrix is basically the matrix which has the microarray intensity in this case, and the sample level and the row metadata are stored in these 2 components of the data frame. And now that I have all this in a standardized format and the data is already downloaded, I'll be able to quickly look at some gene expressions that the paper mentioned are related to B cell differentiation. So just let's run this cell, where I'm basically just subsetting my data matrix for the 3 cells that I am interested in, and using the call metadata to subset for time point at day 7, because that's what I'm interested in looking at, to see the response to vaccine in different patients. And we already have something insightful to look at from the data. So this is day 7, and these are the 2 different vaccines in red and blue. And you can already see that the immune response is different in different patients. Some of them have very high expression of the genes that are related to B cell differentiation, and some of them quite low compared to the other ones. So you can already see, how we could use this data to do phenotyping or like to identify vaccine response profiles. But everyone has seen some form of this data. Transcriptomics data is widely used. But let's look at the process cytometry data that's available as a curated GCT file in Poly. Again, it has the 3 components, which has the data matrix and the associated metadata. And I'm just going to quickly merge the files that I have downloaded. And that was pretty quick. It was, in no time, you have a merged data set. And I am just going to parse the 3 components that we've already seen in our slides. But I just want to go over quickly a little bit in detail about what exactly are we reporting as flow cytometry data. So if we run this cell, you'll be able to see the row IDs are the population name, which are basically the B cell population in different stages. So we have naive B cells, we have memory B cells, and the different columns are the samples at different data points. And the cells itself are cells, the B cells per microliter reported by an analyst. So now you have this whole data frame with so much data easily available to do downstream analysis for vaccine response in healthy patients.
And I just want to highlight this because this is like one of the coolest things I think about the way that you just pulled this data is you mentioned that you had to merge the file. And the reason that we're doing that merge is because each one of those patients are normally stored as individual files, right?
Right.
So what you've done is instead of having to pull individual plots and individual Excel files and comb through and collate that data, we've done it in less than a second. And now we have this fantastic data matrix with all of the sample, all the patient information in one data frame.
Yeah.
Anyway, I just thought it was the coolest thing ever.
No, really, it is quite interesting to see how fast we are able to access the data and merge them. And it's useful when you're trying to derive some insights or hypotheses using publicly available data sets. So I think it's quite convenient to have, such fast access. So we looked at the data matrix, but we also merged the metadata, and we can see, what vaccine was provided and the ethnicity. These were reported at source. But we also have some curated columns. For example, we have the curated disease, which is influenza human. And we only were able to find this data set because it was annotated similarly across geo and the import data set. And that is how I was able to find this data set to start with. So so that's how you would try to find and reuse data sets that already exist in public repositories using Poly. And now that we have the data, again, I want to look at day 7 because the publication mentioned certain characteristic of the vaccine response on day 7. And again, I'm using the count matrix, which is one component of the GCT file, and the column metadata, which is another component of the GCT file. And I just rerun the cell. And now you have a different profile of the patients. These are the cell frequencies. And you can again see there's some variability in immune response in different patients, which we saw in the transcriptomics data set as well, but that was very specific for B cell differentiation. But through this plot, you can see all the different stages of B cell reported for each patient. So there's naive, memory, and transition. So this is, quite useful data, which I didn't know existed before meeting Marianne and, prepping for this demo. But yeah, I think, having this available and accessible through all the public repositories would be great to do, research in AML, immune therapy response, and a lot of applications that we talked about today.
Absolutely. And we are coming near the end of our time together. And we did have just one more slide for you, Marianne, to talk about sort of the future of flow data and where we think it's going. But before we do that, I did just want to talk, thinking about that plot that we just had up, you made a heat map of flow cytometry data across all patients with a bunch of different cell surface markers in fairly easily, like a matter of moments. And it just really highlighted to me the contradiction between when you had the data stored separately and you're looking at the data. I think, Marianne, you used this term very two-dimensionally. When we're looking at the data two-dimensionally, a plot, like how hard would it have been, in your experience to collate the data to a level that you'd be able to produce that kind of heat map, thinking about how flow data used to be versus how we're storing it on Poly and how easy it was to produce that kind of graphic and get those kind of insights.
Typically, we do store the patients individually, and so being able to collate that information either from SCS files or from Excel spreadsheets, and now being able to look at that harmonized and then look at the expression data all at the same time pretty quickly is pretty amazing.
Yeah. So in light of that, and what we have here is just sort of a forward-looking view on the future of flow cytometry data and how it can be, how it's just getting more complex. So these are actually t-SNE plots made from flow data, right?
These are t-SNE plots. And one of the things you said that has happened over time with increasing technology is complexity. And so we have cytometers that are now collecting 38 different parameters at the same time. So, and we have a system called CyTOF that instead of using a fluorescent tag, they add a metal tag and they use mass spec to look at the detection of each antibody or each surface protein. And that now adds a lot of complexity. Typically in flow, we look at things in 2 dimensions. We look at 2 different parameters at the same time and then use a hierarchy to look at different populations. The beauty of adding in bioinformatics into that is really about adding that complexity and stitching it all together. So the human brain can't look at 30 dimensions at the same time, but these beautiful t-SNE plots are now looking at populations of cells. They're each colored differently based on the expression of those markers all at the same time. And here we're looking at a patient with acute myeloid leukemia versus a normal patient's bone marrow. And you can see that the populations do look different. And it gives you a more global aspect on a population expression level. about what is happening in patient samples. And this is, I don't think we're going to stop at 38. The complexity will increase. Cytoph has the capability of doing markers inside the cell and on the surface of the cell. So as we get more complex, there's a lot more information. There's a lot of proteomics that is available to access through flow cytometry. And it's very interesting to see how we'll use that with all the other omics platforms combined together.
Yeah, I mean, absolutely. I think that it's just so interesting, the potential for taking the data that we've collected now, and because it's as it gains complexity and as our tools become more and more sophisticated, our ability to look at the data not just on its own, right? We have a tendency in biomedical research to look at each data type kind of in a little silo, but to do some, inter-omics analysis or some correlation analysis and really lean on the just overwhelming amount of information that can be gained from these data types and look at them in correlation with each other, right? Which is not something that you'd be able to do looking at, one image at a time. There's just no way you can do it with the term my old boss used to use was visual inspection spectroscopy, which I always thought.
And even if you wanted to do multi-omics, it's just like all data is in like different formats. So now you have to learn about what those formats are and how to parse them. So yeah, having multi-omics in research is essential. Only if we could like make it, easy and accessible for reuse. Yeah.